The enteric nervous system, ENS, or intrinsic nervous system, is one of the main divisions of the autonomic nervous system and consists of a mesh-like system of neurons that governs the function of the gastrointestinal tract. It is capable of acting independent of the sympathetic and parasympathetic nervous systems, although it may be influenced by them. The ENS is also called the second brain. It is derived from neural crest cells. Structure. The enteric nervous system in humans consists of some 500 million neurons, including the various types of Dogiel cells, 1/200th of the number of neurons in the brain, 5 times as many as the 100 million neurons in the human spinal cord, and about 2/3 as many as in the whole nervous system of a cat. The enteric nervous system is embedded in the lining of the gastrointestinal system, beginning in the esophagus and extending down to the anus. The neurons of the ENS are collected into 2 types of ganglia, myenteric, Auerbach's, and submucosal, Meissner's, plexuses. Myenteric plexuses are located between the inner and outer layers of the muscularis externa, while submucosal plexuses are located in the submucosa. Function. The ENS is capable of autonomous functions like the coordination of reflexes. Although it receives considerable innervation from the autonomic nervous system, it can and does operate independently of the brain and the spinal cord. Its study is the focus of neurogastroenterology. ENS function can be damaged by ischemia. Transplantation, previously described as a theoretical possibility, has been a clinical reality in the United States since 2011 and is regularly performed at some hospitals. Complexity. The enteric nervous system has been described as a second brain for several reasons. The enteric nervous system can operate autonomously. It normally communicates with the central nervous system, CNS, through the parasympathetic, e.g., via the vagus nerve, and sympathetic, e.g., via the prevertebral ganglia, nervous systems. However, vertebrate studies show that when the vagus nerve is severed, the enteric nervous system continues to function. In vertebrates, the enteric nervous system includes efferent neurons, afferent neurons, and interneurons, all of which make the enteric nervous system capable of carrying reflexes and acting as an integrating center in the absence of CNS input. The sensory neurons report on mechanical and chemical conditions. Through intestinal muscles, the motor neurons control peristalsis and churning of intestinal contents. Other neurons control the secretion of enzymes. The enteric nervous system also makes use of more than 30 neurotransmitters, most of which are identical to the ones found in CNS, such as acetylcholine, dopamine, and serotonin. More than 90% of the body's serotonin lies in the gut, as well as about 50% of the body's dopamine, which is currently being studied to further our understanding of its utility in the brain. The enteric nervous system has the capacity to alter its response depending on such factors as bulk and nutrient composition. In addition, ENS contains support cells which are similar to astroglia of the brain and a diffusion barrier around the capillaries surrounding ganglia which is similar to the blood-brain barrier of cerebral blood vessels.
Please subscribe and thanks for watching.